Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Welcome to your channel, The Anatomy Canvas, with your tutor, Dr. Shehla Aman. In today's class, we are going to discuss with the slides which are present in block A, that is slides of the epithelium, the slides of connective tissue, and slide of the lymphoid system. So now starts. So students, now first slide is taken from the area of the kidney. It is a slide from the kidney, and this is the renal corpuscle, and in this renal corpuscle you are looking at this parietal layer of the Bowman space, which is formed by the simple squamous epithelium. You can see the nuclei of the simple squamous cells which are present, also the nuclei of the simple squamous cell which are present over here, and these simple squamous epithelium lining the parietal space. So this is the Bowman's capsule all over, and parietal layer is formed by the simple squamous epithelium. Also you should note the simple cuboidal epithelial cells which are present over here, which are forming the ascending and convoluted tubules, the proximal and the distal convoluted tubules. So nuclei, you should look at the nuclei. These are the spherical shaped centrally placed nuclei. There are the many sections of the simple cuboidal epithelium. So if this slide is present in your exam, you can write both the simple squamous epithelium as well as the simple cuboidal epithelium. So both these structures are present in the slide. Next slide is again from the kidney, and this is stained with the hematoxylin and eosin section. So this is the slide taken at the renal corpuscle, and this is again the Bowman's space, and this Bowman space, you can see this is lined by the simple squamous epithelium. You can notice the nuclei of the simple squamous epithelial cells which are squamous in shape. Also this is another Bowman space, parietal layer of the Bowman's capsule, which is formed by the simple squamous epithelium. So this is basically slide of the kidney, but for your exams point of view, you have to write down the simple squamous epithelium in this slide. Next slide is the taken from the surface of the simple squamous epithelial cells. So in this you can see these are the squamous shaped cells in which you can see centrally placed nuclei are present. This is the spread taken from the surface of the simple squamous epithelium. Another slide, this is basically slide of an artery, and in which you are looking at the tunica intima, and this tunica intima, you can notice here that this is lined by the simple squamous epithelium, which is here given the name of the endothelium. So look at the nuclei. These are squamous shaped nuclei which are lining the endothelium of this artery. So this is the again another slide for the simple squamous epithelium. Now we move on to simple cuboidal epithelium. This is the slide taken from the thyroid follicles, and these thyroid follicles are lined by the simple cuboidal epithelial cells. So look at these cells. This is the lining of the simple cuboidal one follicle. This one is another follicle, and this is another follicle. So please concentrate at the shape of the nuclei. These nuclei are spherical and centrally placed, and this is a single layer. So this is simple cuboidal epithelium. In the center of this follicle you can see the colloid is present. So these all are the different follicles which are lined by the simple cuboidal epithelium. Again, please remember, some of the students may confuse that this is the slide of the simple cuboidal. This is actually the slide of the simple cuboidal epithelium. This is one follicle, and this is another follicle. Here two follicles are present so close to each other that they give a false appearance of being stratified. This is not actually stratified. This is simple cuboidal epithelium. So please notice and remember, this is simple cuboidal epithelium. This is the slide of the thyroid follicles. This is the higher magnification of the same slide. You can see the thyroid follicles which are lined by simple cuboidal epithelial cells, and in the center of the follicle colloid is present. So look at the cells which are lining the epithelium. These are simple cuboidal in nature with spherical centrally placed nucleus. So this is one follicle and this is the another follicle. So these are the different follicles which are present over here. This is the slide of the simple cuboidal epithelium. So now look at this slide. This is a very beautiful slide, and in this slide you can see this area shows you the epithelium. This whole is the epithelium. So first look at the size of the cell. These cells are columnar in shape, and it is the single layer. And second important point, you should notice that the basally placed elongated nuclei are present. So this is a beautiful slide for simple columnar epithelium, actually the tall columnar epithelium. So this all is lined by the simple columnar epithelium. This is one of the beautiful slide of simple columnar epithelium. So now move on to this slide. Again you can see this is the area where epithelium is present, and in this you can see tall columnar cells are present along with the basally located elongated nuclei. At certain places you can also see the goblet cells are present as well. So this is the slide of the simple columnar epithelium with goblet cell. So you should please mention, if you notice goblet cells are present, so write down the complete name, that is simple squamous epithelium with goblet cells. This is another beautiful slide, and in this you can see this is the epithelium. This whole is epithelium, and this epithelium is a simple columnar again with large number of the goblet cells. You can see the mucus droplets which are present in apical area of the goblet cells. So this is simple squamous epithelium. You can notice the basally placed elongated nuclei of these cells. Also the goblet cells are present. So this is the simple columnar epithelium with goblet cells. Now look at this epithelium. This is the whole thickness of epithelium. First look at this thickness, that this thickness gives you an appearance of being stratified, although it is not. In a clearly stratified epithelium you will be able to see the clear levels of all the cells, but in this slide you can see the nuclei which are present at multiple levels. Also you can notice here large number of the cilia are present, and this line shows you the basal bodies which are forming the cilia. So this is pseudo-stratified columnar ciliated epithelium. Again look at this area, pseudo-stratified columnar ciliated epithelium. Please write down complete name in your OSPE. This is important. This is pseudo-stratified columnar ciliated epithelium. This is actually simple epithelium, but the cells are of a different height. So you can see nuclei at multiple levels, which gives you a false appearance of being stratified. So pseudo-stratified columnar ciliated epithelium. Again another slide for pseudo-stratified columnar ciliated epithelium. This is the special stain. This is the Mason's trichrome stain, and slide is that of the trachea. Another beautiful slide, you should focus at this structure in this slide. So this is the duct of the sweat gland. Slide is basically taken from the skin, and this is the duct of the sweat gland, and here you can clearly see the two levels. The nuclei which are cuboidal in shape are present at the bases. Also the nuclei which are facing the lumen are also cuboidal in shape. You should know whenever we name a slide that it is being stratified, so we give it a name according to the types of the cells which are present on the surface and which are facing the lumen. So here the types of the cells which are present on the surface and these are facing the lumen skin. These are also cuboidal, as well as the basal cells are cuboidal. So this epithelium is a stratified cuboidal epithelium. This is a beautiful slide. This is duct of the sweat gland. Another beautiful slide you can see here. You can see in this duct, this is the simple cuboidal epithelium. This is a small duct. You can see only one layer of the cells with the spherical nuclei. In this area, as the size of the duct increases, the number of the layers lining the duct also increases. So here you can see, especially in this area, two very well defined, very clear layers, two layers. So the epithelium is stratified and the surface cells are cuboidal in shape. They having the spherical centrally placed nucleus. So this is another slide for stratified cuboidal epithelium. This is also another duct. These are the ducts which are present in the salivary gland, and here also you can see the stratified cuboidal epithelium. So these two ducts shows the stratified cuboidal epithelium, and this smaller duct shows you the simple cuboidal epithelium. Another beautiful slide taken from the salivary glands. Here you can see this is the larger size duct. You can clearly see the nuclei which are present at two levels, one which are present at the bases and other which are present at the surface, which are facing the lumen. So the surface cells, the cells which are facing the surface, they are columnar in shape. They have basally elongated nucleus. So that's why this epithelium is given the name of the stratified columnar epithelium. So this is again another slide from the salivary glands. This is the slide from stratified columnar epithelium. This is another slide for the first look at the thickness of the epithelium. So you can see this thickness shows you that these are the multiple layers, means it is stratified. First point you should notice that this is not the simple, this is the stratified epithelium. Second point which is important, you must notice that the surface cells are typically dome shaped. So these are the domes of the cells which are present over here, which I am drawing for you. So these are the dome shaped cells which are forming the surface cells. So this epithelium, which is stratified and having dome shaped cells at the surface, this is the transitional epithelium. So in transitional epithelium, certainly all these cells are dome shaped. Some of the cells are even binucleated as well. So this is the slide probably from the urinary bladder, where epithelium is the transitional. Now another slide. So first remember, please look at the thickness of epithelium. So this is the thickness of epithelium, and this thickness gives you a clue that this epithelium is stratified. This is not simple. Now which type of epithelium is this? So we have to look at the type of the cells which are present on the surface and look at the nuclei which I am highlighting over here. These are squamous shaped nuclei. So first, this is a stratified epithelium. Second, these cells are squamous in shape. So this is a stratified squamous epithelium. Third point which we must know about this epithelium is whether it is keratinized or non-keratinized. So we have to look at the surface. There is no keratin present over here. So this is a very important negative finding, that the epithelium is stratified squamous non-keratinized type. Again another slide, again very beautiful slide with H&E stain. First look at the thickness. The thickness shows that epithelium is stratified, and second look at the types of the cells which are present at the surface having squamous shaped nucleus. So this is stratified squamous epithelium, and also no keratin is present, so it is stratified squamous non-keratinized. Please mention whenever it is keratinized or either non-keratinized. So being non-keratinized is also an important negative finding. So you have to write down that this is stratified squamous non-keratinized type of epithelium. Another slide here you can see this is the thickness of the epithelium. Again thickness show it is stratified. The surface cells, you can look at the surface cells are squamous in shape, and very clearly you can see the keratin which is present over here. So this is the slide which is taken from the thin skin. It is lined by stratified squamous non-keratinized epithelium, and here you can see the dermis, which is the dense irregular connective tissue having large number of the fibers which are present, and these are irregularly arranged. Also you can see this is the section of the hair follicle, and in addition to hair follicle, here you can see a section of the goblet cell, sebaceous gland which is present over here. This is the section of the erector pili muscle, that is the smooth muscle which is present in close relation to the hair follicle. So this is another hair follicle, and this is again another section for the sebaceous gland. This all is the dermis, which is the dense irregular connective tissue. This area is again another section for the hair follicle. This is another section for hair follicle. So you have to write down this slide as thin skin, and for your identification point you have to write the stratified squamous keratinized epithelium along with the dermis and epidermis and the hair follicles and sebaceous glands and etc. So this is the slide for the thin skin. This is again another slide for the thin skin. Here you can see this is the thickness which shows you that the epithelium is basically stratified. So look at the surface cells. These cells are squamous in shape. Also you can see the keratin which is present over here in these areas. Also you can see the hair follicles which are present. This is the section of the smooth muscle cells which is erector pylori over here. This is the section of the duct of the sweat gland, and this whole is the dense irregular connective tissue which is forming the dermis. So this is again another slide for the thin skin. This is the same slide at higher magnification. You can look at this slide. This is epithelium. These are the surface cells, and keratin which is present at certain places. This is stratified squamous keratinized. This epithelium is basically loose connective tissue. So always remember, just beneath the epithelium there is a small amount of loose connective tissue. Look at the cellularity. The number of cells are more as compared to this area. This is dense connective tissue which is the dermis. Here you can see the duct of the sweat gland. Here you can see the hair follicles. Here you can see the smooth muscle which is forming the erector pylori muscle. Another beautiful diagram through the dense irregular connective tissue. So this all is dense irregular connective tissue. You can see large number of fibers which are present in this area, and these fibers are irregularly arranged. In addition to these fibers, you can also see adipocytes which are present over here. So this is a beautiful slide for dense irregular connective tissue. Also remember, please dear student, these slides are taken from your slides of histology lab. Again the same slide for the skin. You can see stratified squamous keratinized epithelium, the hair follicle, the sweat glands, their ducts which are present. This is the structure of the sweat gland. These are the ducts which are lined by the stratified cuboidal epithelium. Here you can see the erector pylori muscle which is smooth muscle in nature. Here again you can see the erector pylori muscle. And this all is the dense irregular connective tissue which is dermis. Again another slide, and here you should differentiate between two types of connective tissue. The one connective tissue which is closely present in relation to this glandular element, and this tissue is basically loose connective tissue. You can differentiate this tissue from this area. This is the Mason's trichrome stain, and in which you can see one type of tissue is this and other is this. So in this area you can see large number of the fibers are present. present, and these fibers are irregularly arranged. So this is dense irregular tissue, and here you can see the density of fiber is less, and in addition large number of cells are present which are forming the connective tissue. So this is a loose connective tissue. So by looking at this diagram, you can differentiate between the dense connective tissue and the loose connective tissue which is present over here. Again, another beautiful slide with trichrome stain. Here you can see this all area is dense irregular connective tissue, and this area which is present in close relation to this blood vessel, this is loose connective tissue. So this is basically the slide for dense irregular connective tissue, as well as you can see beautiful sections of the arteries as well. This is the slide for the reticular connective tissue, and this is the special staining for the reticular tissue, which is the in which you can see these brown color fibers which are present. This is actually slide of the lymphoid follicle, and in this the outer area of the lymphoid follicle is the capsule. Next to the capsule is the capsular sinus, then the cortex, and then the medulla, in which you can see the medullary cords are present. So to this slide you can write down as a slide for the lymphoid follicle, although it is a slide written in your slide box as reticular connective tissue, but this is a beautiful slide for lymph node. So this is the slide of the lymph node. Here you can see the capsule. Just beneath capsule you can see the subcapsular sinus which is present. Here you can see subcapsular sinus is present. Then this area is the cortex, and this whole area is the medulla. So this is the medulla, the cortex, the capsule which is present, and in this medulla you can see medullary cords are present. So this is the beautiful slide for the lymphoid follicles. Sorry, this is a slide for lymph node. Again, the slide of the lymph node at higher magnification. In this again you can see this is the outer area where the cortex is present, and this is the medulla where you can see the cords, medullary cords are present. These are all medullary cords which are present, and this is the again the slide of the lymph node at higher magnification. So students, this is another slide for the lymph node, and this is H&E stained section, and here you can see the outer area is the capsule, and just beneath this light color area is the subcapsular sinus. Here you can see the trabecular sinus which is present, and this trabecular sinus then open into medullary sinuses which are present in medullary cords. In the area of the cortex you can see multiple lymph nodes are present. So these are lymphoid follicles. In the lymph nodes, either the primary follicles or the secondary follicles. Here the germinal center is present. So in this follicle you can see the germinal center is present. This is the secondary nodule. Again in this you can also see the germinal center which is present. This one is primary nodule. So this outer area is the cortex. This is the capsule. Just beneath capsule there is the subcapsular sinus. This is the trabecular sinus. This whole area is the cortex, and this is the medulla where you can see the medullary cords are present. So again another slide for the lymph node, and outer is the capsule. Just beneath capsule you can see the subcapsular sinus. Then this is the trabecular sinus which is present. Then the medullary sinuses and medullary cords. This area is the cortex, and this area is the medulla. So this is another slide for the spleen, and in which you can see two very well defined area. This is one of this area is this which I am highlighting over here, and for other area I take another color, this area. So the first area which I am highlighted over here. This is the white pulp of the spleen, in which you can see the central artery which is present, and this central artery is eccentric in position basically, and the lymphocytes which are surrounding this central artery are forming the periarterial lymphatic sheath. This is again another area showing you the white pulp. These are the sections of the splenic arteries and its branches which are present in the trabeculae. These all the areas are the actually the areas of red pulp which are present over here. So in this red pulp you can see the cords of the Billroth are present, and the sinuses which are present also forming the splenic sinuses. So this area gives a spongy appearance and a reddish appearance in life because it is filled with the RBCs in life. So that's why it is given the name as the red pulp. And this area where the lymphocytes are present, it gives a bluish appearance in life and is called the white pulp. So this is a beautiful slide for the spleen. Another slide for the spleen, and in this slide you can see that this is the area of the this whole is the area of the white pulp. This all is the white pulp. This is again the white pulp which is present, and in the center of the white pulp you can see the central artery which is usually eccentric in position, and in addition to this all area is the red pulp. So you can see very clearly the two areas of the spleen, both the white pulp and the red pulp, and this is a beautiful slide for the spleen with H&E stain section. This is the slide for the palatine tonsils, and in this slide you can see this outer area is the epithelium. So this is the clue to you to identify that this epithelium is a stratified squamous. So in palatine tonsils, epithelium is stratified squamous. Second, you should know this is the crypta magna, which is another identification point for your slide. Third point, you should know that large number of the lymphoid follicles are present. Either they are the secondary follicles or the primary follicles in the present in the tissue. So these 2 points, first epithelium, second large number of the follicle. These 2 points help you to identify the slide. The follicles give you a clue that this is a slide from some of the lymphoid organ, and the epitheliums and the crypta magna help you to identify that this is the palatine tonsil. So this is the slide for the thymus. Here you can see the thymic lobules which are present. These are the different thymic lobules. Outer is cortex, inner is medulla, and in also again first point you should remember that large number of the lymphocytes are present to help you to identify that this is some lymphoid organ. Second point, you should know that these are the thymic lobules which are present, and in thymic lobule the identification point over here is this area which is the thymic corpuscle, which is considered as graveyard of the thymus. Here a little bit keratinization may also be present. So these areas are important from your identification point of view. These are the thymic corpuscles which are present in the medulla of the thymic lobule. So this is the slide for the thymus. This is another slide, a beautiful slide from the connective tissue, that is the adipose tissue. So you can see large number of the adipocytes which are present in this area. This is beautiful slide. The signet ring shape nuclei which is pushed towards the side because of large number of the fat globules which are present. So this is the slide for the adipose tissue. Again, a very beautiful slide for adipose tissue. Large number of the adipocytes are present in this slide. Thank you very much, dear students, for your attention for the today's class. And here I must say thanks to my son Hassan and Shahan, who were present with me in the lab, who were taking the pictures from microscope and helping me to prepare this lecture for you. So thanks Hassan and thanks Shahan for your help, and thank you all dear students for your attention for the today's class. Thank you so much. Allah Hafiz.